Okay, here we are again. I think I'm able to hear myself now. I can see myself also on YouTube. This means we're ready to go. Hi, hello everyone again. Microbehunter here. Welcome to another Saturday live stream. Time passes quickly. It's already been a week. I want to do a couple of experiments today, and I've prepared a few things. I want to show you then what I want to do actually. Usually at the beginning of the live stream, I wait a few minutes until more and more people start to join in. I usually use the first 3 minutes or so just to say hello all around the world. So from Lebanon, from Vienna in Austria, from Belgium, from Rhode Island, US, okay. Bild und Ton arbeiten einwandfrei, so sound and video works without problem. That's really nice. Hello from Connecticut, USA, from Texas, from Portugal. Sound is good because some time ago I actually started to talk and there was actually no sound and video. It even once happened that the video stream was interrupted because of some technical issue. In this case, I had to restart it. So if this should happen, please, give it a couple of minutes and then I might have to restart the whole thing. Hello from the Netherlands. Okay. I hear that today's stream will have some shocking results. Shocking, double meaning here. Yes, I will be playing around a little bit with electricity. Okay, simply to demonstrate a little bit to you how Paramecium react to electricity. There's something called electrotaxis. This basically means that if you apply a voltage, an electrical field across a water sample, then the Paramecium are going to move towards the negative pole. Yeah, and I want to show that to you. Not the first time that I've been doing this. I think last year or so I've already experimented around a little bit. But specifically today I would like to show you some of the things that I experimented around with today in the morning. And not everything was successful. Okay, so the point is that I simply want to share with you my experiences that I have, and then basically you take it from there. Okay, so what I usually do is during the first hour, I simply show you some of the stuff that basically I want to show you. Okay, you can of course post questions in the chat. If the questions are off topic, then after 1 hour I will answer them separately. But if the questions are on topic, then I will try to answer them as we go along. Hello from Paris, I just see here. Okay, no echo, that's also good. Okay, yeah, so every now and then I will interrupt myself a little bit and simply to see if, what's written over here. If you have a specific question for me, then please indicate this by writing at Microbehunter or at Oliver. Oliver is my first name, so it's easier for me to filter the messages a little bit. Yeah, so I don't know where to start because there's so many things to talk about. Okay, so I'm going to start as follows. What I've got over here is a water sample, and I'm going to take it step by step. And this is going to be our source for Paramecium. Paramecium are single-celled microorganisms, and this is a regular pond water sample. Just one of the previous videos that I made and published actually shows how I collected this sample. And if you look very carefully, you're going to see that there's this white slimy stuff in here. Okay, these are so-called wheat grains or cereals, so to say. I wonder if I've got some of them left over here. Just a second, I'll show them to you. Okay, here they are. Okay, they look like this. I've got some, that's the food that I use. That's some dry yeast. That's the powder. Okay, I'm not using that, but I'm using those wheat grains here. I'm just going to pick out one of those. It's just a cereal grain. Yeah. Over here, which I'm using to grow Paramecium. And this thing over here, that's another food source. These are compressed algae tablets. Some companies sell them as some kind of food additive. I don't eat them, but I take a little bit of this and to feed my water fleas. Yeah, so I don't need those. But for actually feeding Paramecium, what I do is I take a wheat grain. Like this, and I crush it between 2 spoons, and then I drop it into the water sample, and then you have to wait for approximately 1 week, and then it's going to turn all slimy and soft, and this is basically a sign that there are a lot of Paramecium growing on here. So this will be our source of Paramecium. When you do stuff like this, please be careful. You are also growing many bacteria. Okay, so you always want to make sure that you stay hygienic. This basically means try to avoid getting concentrated stuff on your fingers. Okay, so For feeding Paramecium, I guess anything with starch would work. Yes, generally yes, but those wheat grains have an advantage, and I'm going to show that to you, is that I can easily pick it out. Okay, so that is actually it's more the physical consistency. If I just drop in a whole bunch of starch or other food into the water sample, there's the real danger also. I wouldn't say danger, but the real problem that bacteria are going to grow so fast that the Paramecium cannot keep up. Because what happens is if you add the wheat grain, then bacteria will feed on the starch and the Paramecium will feed on the bacteria. So it's a tiny food chain, and it's really practical to use those wheat grains. You crush them first to open them up, and then it's easier to pick them out. Okay, so that will be our source of Paramecium. Okay, so that's number one. So it's just a little some background information so that you understand where I get it from. So they're really easy to grow. Yeah, again, I made already a whole bunch of videos relating to this, so you might want to check them out. And then now basically what I have here is, and I will not be using this, but I also just wanted to show you, what I want to do is I want to put them on the slide and I want to apply a voltage. So I've got some cables here. I'm going to show you also the power supply unit that I have here. And so they are actually selling also those things here, and you see over here there's a little, how do you say, channel, and if you add some liquid in here, then you and you're pulling away liquid in here, then you can actually see microorganisms streaming across here, and you can actually put this directly under the microscope. Problem with that is that those little containers for liquid, which basically connect, the two containers are connected over this little channel over here. They're a little too large, and sometimes they're in the way of the microscope objectives. So that's a little bit of a very good solution for observing this, but also a very clumsy solution in the sense that you cannot work with all magnifications because it's simply too large, right? And this comes along with a pipette. So for example, you can put this in here, and then you have this one over here, right? And then you can essentially push some water through it. You can flush it out again and clean it out, and then there are also those, So that's basically the concept of a so-called of a micro aquarium, right? And I've also experimented with that. I put some wires in here. It also kind of worked. But the thing is the point of this channel and this live stream is to experiment with things that everyone's able to do, right? And not everyone has this, and you've got to buy it, and you have to order it, and so you actually want to experiment around with stuff that you have available at home, right? So that's basically what I would like to do today. So I'm not going to be using this here. This is a so-called a micro aquarium system. It's called MicroLife. I just want to share it with you because this actually, I've also experimented with that. There's an instruction manual in here as well. And so that's you get the idea. So if you're interested, that's the company, right? Who manufactured this. So, but I would say that has a fairly specific uses, and because it's rather specific, I think most of you will probably not be using this. And I was therefore playing around with a couple of other ways on how you can make let me put it somewhere where it doesn't break. I've been playing around with other possibilities of connecting electrodes. Okay, and you can already see some of them here, and I will explain how I've made those. They are over-engineered. It's more simpler, okay? I will just show you, So, a couple of questions here, again. The metal strips on your slides look like a piece of Matrizenband. What is used by dentists? What do you use here? The metal strips here? You're gonna laugh. The question is what are these metal strips? Or the one over here? Almost embarrassing. It's a tin can, okay? Yeah, it's for a beverage. From a Coke can. So that's it. Okay, so I simply used scissors to cut metal strips here. So it's aluminum, which it does conduct electricity. So as you can see, absolutely not high tech. Okay, absolutely not high tech, deliberately not. I wanted to use everyday materials here. Okay, so that is, yeah, is this some kind of an electrophoresis chamber? No, it's not. This one over here. Well, actually, it's not an electrophoresis chamber, but I understand the similarity, because in an electrophoresis chamber you also apply voltage, but actually that's it's a micro aquarium. Okay, that's at least the way that it's being sold. Yeah, so. So what I'm going to show you now is my first attempts here. Okay, and I'm going to show you also of what I've done here. So what I've done here is I've glued using some nail polish, just regular nail polish. This one is already pretty dried up. I've got another one somewhere, and I don't even know where to put the bottle. My desk is so extremely messy. I've actually problems finding the things. Okay, but yeah, but you get the idea. Where did I put it? Did I already? Oh, I put it back into the into my cupboard. Just a second. Here we go. Okay, yeah, yeah. So I just used regular nail polish. This one over here, you can see, is already pretty dried up. So you have to add some more acetone. And what I've done is I've taken those cover glasses. Yeah, so those cover glasses, and look, these are pretty huge. These are very large cover glasses, yeah. Yeah. And what I've done now is I've cut some strips of the cover glass, and using the nail polish, I glued them on here to make some kind of a channel, yeah. And then on the side here, I glued some of those tin can metal strips. And then the idea kind of was, and that's why I'm saying, oops, it's a little bit over-engineered, the idea is I put the sample in here, I connect the voltage to those two metal strips, and a cover glass goes on top here, and then I can actually see the Paramecium, they move towards the negative pole. It's called electrotaxis. That's kind of was kind of the idea, right? However, I found some problems with this system, but I'm going to show you first what I have done in order to cut those cover glasses, okay? Just want to show this to you as well, simply because even if you're not going to be making this type of chamber here, maybe the thing that I'm going to show you might prove useful anyway, okay? So what I'm going to do is the following. What I have here is I just want to show this to you. Maybe you know these type of tools here. These are the grinders. So when you put this in here, then it spins very quickly, yeah, and you can actually grind something on glass. Yeah. So actually, you could actually also use this to. I tried it to kind of label glass in microscope slide. That would actually work. Yeah, and. However, I'm not going to be using this the motorized grinder. All I need is this tip here, and this tip contains basically diamonds. Okay, and I just want to show you those diamonds here. These are industrial diamonds. So I'm going to also put this under the microscope now. So simply that you see how this looks like. Let me quickly check the chat again. Okay. Yep. Okay. So let me show you the stereo view. Yes, here we go. I just want to show you the tip of this here. Here it is. Ah, that's nice. Let's zoom in a little bit. I have to focus. And you see those tiny little, it looks a little bit like grains. Where is this? Here's the arrow. Yeah. So those little things that you see here on this round tip, I think these must be diamonds, industrial diamonds. Very hard. So basically much harder than the glass, and therefore you're able to grind the glass, and you're able to scratch it. Okay. So that's kind of the thing what I've been using. And then what I did is the following. Let me go back to the desk view again. And I'm going to give you a short demonstration. And I just realized that maybe the surface is not the best because it's soft. So that's actually not a good thing. So what I'll be using is, okay, I'll just be using this one over here. This inverted, how do you call this, Petri dish as a surface. I put this, this cover glass on top here. I need something straight, like a ruler. I don't have a ruler, so I'm going to use another Yeah, another slide. It's very difficult to see, of course, because it's transparent, right? But essentially, I'm just using this slide now as and then I use this tip here, and I'll carefully, yeah, scratch across it a couple of times. I mean, actually, I realized you don't even need a lot of pressure to do that. Yeah, you will actually feel when, yeah. I mean, I don't know if you're able to even hear it or not. Yeah, and then sometimes it will separate on its own, or when it's scratched, you just give it a little flick. Okay, you see, almost. Yeah. Yeah, so I didn't scratch it properly all the way, but that is how I made those little strips of cover glass. Actually, yeah, I really didn't properly. Yeah, now it works. Let me try this again, just for the sake of demonstration, because now I actually want to try it again. That's again a little bit unfortunate that you're not able to see it because of the lack of contrast. Yeah. Yep. Here we go. Let's try it again. Okay. I don't know if this, and yes, now it's separated nicely. Do you get the idea? Okay. And that's basically how I made those. Yeah, those strips here. And then I'm also just going to demonstrate this here on one of them, so that you see what I have done. And yeah, I've simply taken then the how do you call this, the nail polish. You probably want to remove and clean this first, so that there are no glass pieces here, because otherwise it's not going to be flat. Let me put this here away. And yeah, and then I'm going to show you what I think is the better method. Why am I showing you then, if there's a better one, why am I showing you this? Because yeah, because that's what I've been doing today in the morning, experimenting, and maybe it will actually, how do you say, maybe there is a use of this. Yeah, if it's a little bit better optimized. You haven't seen this now because I was kind of holding it away. Yeah, so I'm just applying some of the. Now, why nail polish? The reason is because nail polish is not water soluble. So that is the, yeah. And then I simply glued it on here. I pressed down a little bit. Yeah, and I put it on a hot plate, my coffee warmer, for those of you who remember, or you can just wait a couple of hours for the whole thing to dry. Yeah? So that's basically what I've done, and I've done something similar over here with the metal strips from the beverage drinking can. Simply used I used the nail polish to glue it. Yeah. The disadvantage of the nail polish is, you can see it here, as it dries, it starts to make those bubbles. Not a big problem, but of course it could be that some of the specimen is probably sucked in beneath here, right? But that's the way that I've done it. So nail polish has the advantage, it's not water soluble. So if you put a water specimen on here, then it's not a problem. And the second thing is that after it's dried, it sticks really well to the glass. Yeah, so these are the reasons, and yeah, and it dries pretty quickly as well. Right? And I've used the blue one because it's easy to see. Of course, you can also use a clear nail polish. That's a question of taste, really. Okay, so let me go a couple of Is that a slide or a cover glass you cut? I cut the cover glass. Okay, so again, what I've done is and it's difficult to see. Yeah, I took a cover glass. Yeah, over here. Let me do this again. Yeah, and basically I used this diamond grinder cutter. Let me move this a little bit over here. Yeah, to cut across. By the way, I still do need that because look, the other one that's actually much easier. Why not just do it like this? Yeah, look here. That's the beverage can, and I just used, how do you call it, nail polish again to directly glue it on here. And then I realized that the other cover glasses that I have over here, okay, were sometimes a little too small to fit over. And the large one over here, it did fit over, but I felt that it was too large. And the reason is because I was kind of worried that if I apply a specimen over here right now, because you see there's such a big overlap between the glass and the electrode, I was kind of worried that maybe some of the specimen is also going to be sucked directly over the electrode, and yeah, but I want the specimen to be directly here in the middle, right? So what I'm doing right now is I'm just going to take this huge cover glass here, and I'm also going to take off a little edge, but this, right now, yeah, across, simply to make it a little bit shorter. Yeah, so over here like this, then I take this here again, and again this, how do you say, is only for to have a hard surface, because the table down here, this is very soft plastic. Yeah? So, and then I'm just going to go like this a couple of times, and you turn it a little bit until one of those diamonds will actually start to scratch the surface. Yeah, and you can actually feel it. Yeah, and then once it's scratched, oh, it already separated. Okay. Yeah, almost scratched the petri dish as well. Yeah, you see it already separated, and now I've got this also in the correct size, so that I can put it directly over here. Okay. Yeah, so that is basically the system that I'll be using, because this system over here, which I tried out before, does have a little problem. Okay, and I just wanted to show that problem to you by putting it under my stereo microscope. So where is the stereoscope? Here it is. Where's the picture coming? Yes, just want to show this to you, because this is a little bit of a, let's go out with the magnification again. Let's focus it again. Okay, so you see over here there is one electrode, right? And over here there is the other electrode. And now I want to explain something to you. Do you actually see this bubble that you have forming here, right? This was actually an air bubble. But when you connect it to the electricity, then there will be electrolysis happening. This means that the oxygen and hydrogen gas will be formed, and there is not enough possibility for the gas to escape. So that's a little bit of a problem, right? That's, yeah, and if there is too much gas here, then there will not be any contact anymore with the liquid. You actually see that there is stuff moving over here, right? Yeah. There are a whole bunch of ciliates in here, right? So this is a little bit the problem that I discovered afterwards, that there is, it does not allow the gas to escape, and therefore sooner or later it's going to lose connections, because as you're going to see, there will be tiny bubbles forming here as the water is split up into oxygen and hydrogen. Yeah. PVA glue would also work as a glue, wouldn't it? I deliberately did not use PVA glue, because PVA glue is water soluble. Okay, I mean, I know some glues that are water soluble, but once they're hard, they won't dissolve anymore. But I'm not quite so sure about PVA glue. Yeah? So this is the reason why I've used nail polish, but I'm not saying that this is the best type of glue to use. You might also try to play around with silicone. Maybe there are those the silicone that is used for sealing, off some corners and edges might also work. So I just wanted to show this now to you also now under my compound microscope. Let me quickly show this. Let's go here. Let's see if this works. Yes, okay. So let's remove the filter. And I'm going to now put this here. This is now at low magnification. Okay, a little too bright. Yeah, you see over here, that's the basically the place where the cover glass is glued, and you can actually see that some tiny cells, here it is, some tiny cells actually also managed to find their way here. Yeah, you can also see some movement into those air bubbles. Yeah, it doesn't matter. We have to look at the cells that are in the canal, in the little channel here, right? And we see over here indeed a whole bunch of Paramecium from before, right? But the problem, as I said already, is that over here, yeah, just move over. Where is this? Here we are. Look. Yeah, this is basically an air bubble, air bubble, air bubble, air bubble, air bubble, air bubble, and this is the metal electrode, right? So there is no connection anymore. Look how many, wow, didn't expect this either. Look how many Paramecium there are here. Yeah, I suppose they are all going to the air bubble because they're running low of oxygen, so they are of course going there where there is lots of air. Okay, yeah. So but you see, that's a little bit the problem with this system here. Okay, a couple of comments here. Do you know any substitution for U-Pearl that is compatible with alcohol? Oh, no, I don't. That's unfortunate because U-Pearl, maybe there is. Yeah, but I'll talk about this later. It's a little bit off topic. Okay. Okay. Very good. Thank you very much for this info here, because we just talked about actually using PVA glue. I've tried to dissolve PVA glue in water to redo a permanent mount. Even after a week in water, it does not dissolve anymore. That mount is pretty permanent, I would say. That's very good to know, thank you. However, it could also be that, I don't know, because once it's dry, then, yeah, I'm going to make an experiment. Maybe I'm just going to let some PVA glue dry directly on a slide without cover glass, and then I'm going to try to put it into water to see if it actually dissolves. Because if there's a cover glass on top, then the water might not be able to reach all of the parts directly. But then again, yeah, after 1 week, that should be more than enough. Okay, so this is basically, this was not a background info. Okay, I have to look at the time a little bit. Yeah. So yes, let me go back to the desk view. Okay, so what I'll be doing now is I'll be trying to use this one over here now to put some of the Paramecium on the slide here, and then I'm going to now show you my power supply unit and how I'm going to connect this. Okay, I need to do a little bit of cleanup first because there's so much stuff on my table here. Yeah. So let's, I'm going to now flip over. I have to go to the different view now. And look, there are now two images. There is the microscope over here, and I want you to have a look at the other one next to it that I put there, because that is my desk view. And look what I'm doing now. I'm just flipping this over now, and I'm turning it. And over here you see my power supply unit, which I'm going to turn on. Okay. So, and on the bottom of the power supply unit, I've got a plus and a minus, and this goes to those cables here, right? You know what, I'm just going to leave it here. I'm going to go back to the desk again. Here we go. Oh, okay, that's my, yeah, these are the cables. It's upside down. Okay, and then I will be connecting it over here, like this. Yeah, one on the left side, one on the right side, hoping that the the glue, the nail polish is strong enough that the aluminum is not ripped off. Yeah, so that's kind of the strategy. And because I only have one camera, unfortunately, I have to always keep on flipping it back and forth, so that you're also able to see it like this. Okay. Yeah. So it works like this. Over here you see the voltage 8.2 volts, and I can turn this knob over here. Now nothing is connected, and I can go up all the way to 30 volts almost. That's gonna kill the Paramecium, I found out. They don't like that. So I have to make sure that I kind of stay at a fairly low voltage. That's for the coarse adjustment, and this the bottom knob here is for adjusting it more finely. Not really needed, I would say. Yeah? I'll just stay with the coarse voltage adjustment here. Maybe I'm going to go up maximum 10 volts. I don't know. Yeah. Okay. Yes. Okay. Again, something about the PVA glue, which is quite interesting. So let's give it a try. Okay. It's always like this that there's always a little bit of an uncertainty because it's. Those live streams are a little bit improvised. So, and let's flip it straight again. Here we are. And number one, when you do anything on your work table, the first thing that you want to do is you want to make sure that everything's properly cleaned up and that you only have those things lying around that are actually needed. Otherwise, there's too much destruction going on. And I'm going to remove those here because I won't be using those. Yeah, but maybe it still proves. By the way, this method might still also prove useful if you want to make some kind of spacers, for example, for observing sand samples or other things. Yeah, so that's the reason why I still wanted to show you how to cut those cover glasses. Yeah, see, Yeah, it's actually quite a good cut. Yeah, and over here I've got let me trash this. I've got now the cover glasses here which fit across here, so there is a little bit of an overlap with the electrode, but not too much. So if there's some water or some sample that spills over onto the electrode, which is not a problem, at least not too much of it will be on the electrode. Yeah, so most of it will still be here in the center, I hope. Yeah, so this is basically step number one. And now I have to do the following. I'm going to now prepare. I have prepared and I cannot find it again because, oh, here it is. I'm going to prepare a little bit of concentrated alcohol. Okay, number one. Okay, number two. I need the following. I need, where is my, the other thing? Just a second. I need this here. This is a lighter. Okay, I will tell you why, because I need to disinfect something here, because safety is also important, especially when we're messing around with sharp edges. You don't want to hurt yourself, especially when you are then playing around with microorganisms. Not a good idea. So what I'm going to do now is I'm going to do the following. I'm going to carefully take this here, and there will be many Paramecium and other ciliates around the slimy layer of the wheat grain. Okay, so that is the thing. So what I have to do is I have to pick out this wheat grain here carefully, and I will simply dip it on here. Okay, and I'm also going to take the other one because maybe there are more on here. I don't know. Yeah, because each one of these wheat grains is a little bit like its own tiny little ecosystem. The third one here as well. And let's give it a little bit of a, let's rinse them out a little bit. So this goes back. So now I'll put this away again. I put a lid on top. Yeah, just to prevent it from spilling and to prevent too much water evaporation. And look what I'm now doing next. This is the alcohol. Look how pointed they are. If I hurt myself, I don't want to do that because I'll be transferring microorganisms, bacteria. I'm not so much worried about the ciliates, the Paramecium, more worried about the other unknown bacteria that are in there. So let's simply disinfect it. And back in the day when I was still working in the microbiology lab, what we did then is the following. You want to remove the alcohol as well. Yeah, and you simply burn it off. Okay, and now basically it's sterile. Okay, you got to be a little careful. You don't want to have a flame right next to the other alcohol here, right? You don't want to overheat it because otherwise you're damaging the metal. Yeah, but right now, now it's totally, fine. Yeah. So let's put this away so that I don't knock it over either. And now we do the following. We're going to have a first look at this under the microscope, because we're not quite ready yet. Okay, here we go. This is working. So without cover glass, without anything, at low magnification only. Okay, we're just going to do a very quick check. If we're able to see some movement. Okay. Yeah, there are a whole bunch of ciliates. Not so many larger Paramecium, I think. Ah, no, there are. Here they are. Okay, you see them? But what else do you see? You see a lot of these white dots. These are starch grains from the from the cereal, right? And many of these Paramecium are still clustered around here. So what I need to do is I need to break this up a little bit, and I need to spread it a little bit. So what I'm going to do now, therefore, is I'm going to take it off again here. We're going to go back to the desk view. I need a pipette. Okay. Over here is my pipette. And what I'm doing now is I'm simply going to pull it up and down again, simply to break up any clusters that there are here. Okay, and I just realized that there is not enough liquid here either. So I'll be adding simply a little bit more water. Okay. To make sure that there is enough liquid present. And let's resuspend again. Because if there are clusters, then the Paramecium will simply stick together around the starch and the food. And I just found this to be a little bit better, and also better for visibility, because then the starch grains also don't cluster, don't form these larger clusters either. So the question is now, I don't know if there's now enough liquid here or not. Let's add a little bit more. Okay, I think that's enough. This one goes into the trash can. I've got a separate trash can over there. And then let's drop the cover glass on top. And now we have to really make sure that there are no bubbles next to the electrode. So, and that's fine. Okay. Yeah. We see that there is a little bit of water now also on the electrode here, on here. That's fine. So next thing that we do is I'm just going to put it directly under the microscope again, and let's have a look how it looks like. So I need to change over. Where is the scope view? Here we go. Still so many starch grains. I don't know. Yeah, and occasionally those larger ciliates here, the Paramecium here. There is a bubble here, but that's not a problem. So I do hope that those starch grains do not really inhibit the electricity too much. Here's another one. I'm really more interested in those larger Paramecium. So it might be a little bit difficult to chase them, but maybe we can see how they will start to accumulate on the negative side of the electrode. Yeah. I tried this before, and they reached the negative pole of the electrode, and then they died. They started acting up strangely and changing their shape. Some of them popped open, so evidently the voltage was too high. There is no voltage yet. Okay. So we might also observe a little bit how the smaller cells, what they do and how they react. Yeah. Maybe I'm going to go down with the magnification. Let's take the 4 times. Maybe that's better. Let's just stay with the 4 times magnification, because we're not so interested in the actual shape of the cells, but more in the general movement. So let's put the arrow away. Just put the arrow away. Here it is. Okay, so I'm now going to, I have to turn this around. Now look at the top. Here we go. Here is the power supply unit. Now it's 0 volts. I have to now connect. I have to connect now the electrodes. So here, right? Just put one of them on here on one side. I really hope that this actually works. I ripped this. No, it's fine. It's fine. It's fine. Okay. I have to connect the other one. Okay, just a second. The cable is a little too heavy. Okay, but now it seems to be stable. Seems to be stable. Yeah, it seems to be okay. So what I'm gonna do now is on here. You're just not able to see the clips, yeah. But this one over here is the black one. That's the negative pole. This means that the Paramecium should be moving towards the left side here, but because everything is flipped in a microscope, they should be going to the right side. Okay, that's kind of the idea. So what I'm gonna do now is if you now look at the power supply on top here, right? I'm now carefully going to turn up the voltage. There's some movement going on. What's going on here? I know why. Because the wire pulled on the thing here. So what I have to do, that's not, I have to hold the cable a little bit. Or I will try I have to hold the cable a little bit. So, and I'm going to now carefully go up with the voltage. Hoping that they're going to move now, they will start moving now to one side. The density is not quite as And I will go there now and I will see and wait if they are going to slowly start arriving there. Because that's kind of the idea. At least it worked just before. I don't know if 6 volts might be enough or not, but I had all of them accumulate there. Now see, this guy is not really going anywhere yet. A little bit undecided. So maybe I'm going to go up with the voltage. Maybe there is a critical voltage. Let me quickly check the edge here. You see that there are bubbles forming here. So this means there is contact with the water. It would be. So when are they coming? Oh, I had the cables flipped. Just a second. Are they going to the other direction because I flipped the cables? That's the reason. Okay, my mistake. Okay, I had them. I've got black and red wrong connected down here because I tried to change the yeah. Yep, that's why they're now. Okay, I have to redo this. I will tell you why. Okay, let's flip this. Take this down here because I messed up here at the top. Okay, let's try it again. And unfortunately, also the thing now separated the glue. This was the problem with the demonstration effect. So let's try it again here. See, the problem is that the thing here. Let me. As you can see that. See, this is kind of unfortunately it's separated here. Okay, so what I have to do is I have to somehow make sure that this is a little bit more stable. Okay, so maybe yeah, let me try it again. I think I already know what I'm gonna do. I think I'm going to make a proper video because every time when this is done live, you never know if it's gonna work the way it's supposed to work. But we've seen now that evidently the glue didn't really hold properly. There is now also, yeah, we still have enough liquid in here, I hope, or not. Let me see. Yep, I think there's still enough liquid in here. Let me check. Here we go again. So, number 1, let's do this properly. Number 1, we flip the cables the way it should be. Okay, that's number 1. So, number 2, I think I'm not going to clamp them on, I'm just going to try to hold it on here. Okay, that might also be a thing. And I'm going to go up directly with the voltage so that because I only don't have one hand here. So this basically means that I'm going to go to this part here already. And this is the place. Let me see. Are there still some cells in here? Paramecium here, yes. There are still some occasional ones floating around. Not enough in my view. There are. Maybe the electricity already did the rest to them. I will try the following. I will just try to wait here in any case. Maybe a few of them are going to arrive. I don't know. Okay, let's try this. You see the bubbles forming? Nope. Okay, people, I think this needs to be optimized, but you see that there is a lot of electrolysis happening here. Okay, this is also a thing where I just have to say this is not really working the way I hoped it to work, which is a pity, but I'm not going to give up. I'm not going to give up, and I'm going to try it the last time. Yeah, so pins to fix the contacts, that could be a possibility. There's some clamps as well. So let me again have just a second, the desk view here. So the problem that I have is that, of course, when there is the clamp connected, it kind of lifts up the whole thing here, right? So this is therefore not an ideal solution. But what I can try to do in any case, I'm going to then still try it now with those here. Why not? Yeah. So let me put this away. As a so to say, as a last try. Otherwise, I tell you the following: I will try to optimize this a little bit, and then maybe I'm gonna make a separate video. So let me quickly disinfect it again. Yeah, I mean, I've got some of this stuff already on my fingers anyway, so I have to wash my hands later. I'm gonna add a little bit more to make sure that there is no air bubbles. And yeah, where is the other one here? Here it is. Let's drop the cover glass on top here. No, I'm going to use a larger cover glass, a full-size cover glass. Yes, there seems to be good contact now. So let's see how this works. I don't know, there are now extremely few Paramecium visible. I don't know where they all are. Here's one, two. Dang, I don't know where they all are. For whatever reason, maybe I already washed them out because I kept on picking. Here's one. Here's another one. Yeah, there are a few. Maybe a total of, here are a few more. Yeah, here more. So what I will do is the following. I will reduce the electricity a little bit, the voltage a little bit, because maybe it was a little too much before. That's why they all died. And I will try now the following. I will try to carefully connect. You know what I'm going to do is I'm going to do the following. I've got one extra cable here. And the other one I'm going to hold here now. And let's hope that this works. No, the slightest movement is already a problem. Ah! Did I lift it? Yeah. Nope. Just a second. This is, ladies and gentlemen, I have to admit that today's demonstration is almost nearly a complete failure. Because I'm kind of worried that there is now a little bit of an air bubble. Okay. I think I'm going to try something different now as a last emergency resort. We're going to do the following. I show you what happened. I'm still not giving up. Where is this desk view? Here we go. Okay. Again, look, I ripped it off the electrode. So we're gonna do the following, people. Where is it? I'm going to try it with this one here. And I'm going to now use, and if this doesn't work, then folks, then we're just going to say that not all of the things actually work the way they should work. Number one, number two. I fold it over. I put it in here. Why don't you just do away with the aluminum strips, just attach the cover glass to the slide with the clips? Yeah, I don't know if this is gonna work. No, I can't, because if I just the suggestion was the following, to kind of just attach it like this, right? But the problem then is that the slide is not going to be flat on the stage, because yeah, it's going to be also here on the bottom. Yeah? So this is going to be now the last how do you say, hope for me to make this work. I'm just going to make those electrodes like this. If I'm able to actually get it in here. Okay. So, like this. Like this. Okay. So, maybe this is this is gonna work. I have to now do the following. I have to hope that this actually will give me enough of these cells in here. So, same thing as before. No, I'm going to do the following. I'm going to mix them here. And where is this? So let's put it in here. Okay. See, all of the starch here is a little bit of an issue. So, and I'm going to now add more water. And actually need a large pipette. So I'm just going to use this. To resuspend. And I'm a little bit worried now that there might not be enough. Paramecium in there. So I'm going to do it differently now. There is now an air bubble in here. What they said is yep, now we've got it filled. So, because the liquid has to run through. Okay. Okay, this one is now pretty full, the other one not. Okay, just a second. Or is it also... Ah, no, both are... Is this full already or not? I have a problem seeing this. Okay, no. So... I really have to disinfect everything afterwards with alcohol because this is a real mess what I'm doing here. You can't see it. So I think that is enough. So I'm going to give it a little cleaning. I see that there is an air bubble in here, which is also not nice. So, and for this reason, in order to remove that air bubble, I'm going to take this one here. Yep, and the bubble is gone. Very good, very nice. Okay, so let's try. I don't know. Today is indeed a little bit of a highly experimental. How do you say? Yeah, it's highly experimental. Let's see if I'm able to see something here. Scope. Yeah, that's how it looks like. See, and you can actually see that over here, on the left side here, it's kind of blocking. The objective is kind of blocking yeah. Yeah. So that's a little bit the problem. But how am I going to do that? Maybe, I don't know if rotating, no, it doesn't work. Yeah. So that's a little bit the issue that I'm having. You know what? I have another idea. Let me try it on my stereo microscope. Stereo. Yeah, see, but the Paramecium are too difficult to see here. Yeah, that's the problem. Huh? So let's go again over here to the regular view. Okay. So here we go. So that's gonna be my last try, honestly. So I'll insert the electrodes now, one on the left, one on the right. So this is indeed a little bit more stable. And let's have a try. Okay, so over here. And now on the other side. I know, let's do it, let's reverse it because they will move towards the negative pole. And let's see. Okay. If they are moving, yeah. If we see an accumulation of Paramecium here and other ciliates on this side over here. If not, then I'm completely frustrated. But it did work before. Okay. But look, the smaller ones are actually, thank you for the questions. Look, but the smaller ones are indeed moving all to one side here. They're moving to the, look at this. That's actually, the Paramecium should be moving to the right and the others are moving to the left. That's interesting. Okay, it flipped because of the weight of the, but actually, so let's try it again. So just a second. Yeah. Oh, by the way, thank you for the generous donation. But look, they are moving. Okay, cool. You know what? Okay, I'm not gonna touch this now. I'm going to I'm not going to touch just a second. I just want to show you. It's the wrong scope view. So that's the one I wanted to show you here. We're now at 4.5 volts. And these are not Paramecium, these are the ciliates, but that's fine. I'm going to try now the following. I'm going to try now to reverse the polarity here. I really hope that this is going to work, that they're going to move towards the right, to the other direction. Now they're disconnected. They're disconnected now, they're moving random, and now let's see if they're moving in the other way. I really hope. Are they moving now the other way or not? I think they are now starting to move to the right. Yes. Okay, at least that seemed to have worked. See now that those tiny cells are moving now to the right. Not enough Paramecium in here to, yeah, but they seem to be moving into the opposite direction as the Paramecium. Because the Paramecium move towards the negative pole and they seem to be moving towards the positive pole. Yeah. Okay, let's do this again. Now I'm slowly getting excited. I'm getting excited. Now I'm gonna reverse it again. Let's see if those cells are moving then again to the left. Okay, let's try it again. Look what I'm doing here on the bottom. And the other direction. Well, I tell you, I'm a little bit relieved. Now I hope that they're moving again. This way? Oh wow! Yes, they're starting to move again into the other direction. Wow. Okay. I'm relieved that I'm at least able to demonstrate this to you. It's not super. I know it's probably not the most exciting of things, but yeah. Let me see. Let's add a little bit of color. Nah, who cares? No, forget it. It's just yeah. Yep. Now they're moving again. Here they are. Whole swarm. So I'll just use this time now to go back to some of the questions in the comments here. Oh my gosh. You're dealing with very small currents here. You might be able to get away with a very fine transformer wire. Okay. Yeah. You could take the rubber insulators off the clips and sellotape the clips. That was also a possibility, yeah. Yeah. Wrapping the tape. So thank you again very much for the generous donation here. I will use always those donations to buy microscope slides that I'm going to show you. Yeah. Failing is part of science. Yes. Yeah. So I think that the next interesting, I think the next interesting thing here would be to observe if different types of cells move actually into different directions. Yeah. What happens here is that those smaller cells that you see here in the background, they are moving now towards over here in this on screen. They're moving to the left, but that would be the positive pole. That's interesting. So they seem to be, yeah, so because Paramecium normally move towards the negative pole. Yeah. So for those of you who just joined in, I'm applying now a voltage to a water sample after trying for a long time and not being able to manage this. I'm applying, and by changing the voltage and the polarity, you're able to make the cells move. Yeah. So they were now moving to the left. I'm going to reverse it again down here. And maybe I'm able to make them move now to the right again. Maybe I'm going to go up now. Now I'm a little bit more brave. Let's go up a little bit with the voltage. I'm almost at 7 volts. Yep, and now they're moving again to the right. But others, larger ones, seem to be moving more to the left. Look, the larger ones are moving to the left and the smaller ones to the right. I'm going up yet a little bit more. If I go up too high, Can you show up where they all ended up? The thing is the following. Because of those little tubes on the side, I'm limited in the movement. That's a little bit the problem, right? Yeah, so what I can, yeah, the slide flipped out again. Yeah. Because I cannot move further because I'm bumping into the objectives. Yeah. Yeah, I'm already limited here, yeah, in the movement. So that is indeed one of the problems with this setup, that I'm not able to look at all of the parts. And under the stereo microscope, I would not be able to see those cells. Yeah? So I'm already limited in the movement. That's the problem. Yeah? Yeah, the weight of the... So this was now a water stream. Okay. Okay, I think, what time is it? Okay, it's already over 1 hour. You know what, just for the last time, I'm going to reverse the polarity now, carefully. Again. And let's see if they turn around again and start moving to the left. Yeah, the larger ones move into the opposite direction compared to the smaller ones. Yeah, interesting. Okay, didn't expect that. Let's go up a little bit with the voltage. Maybe I can increase this. Yep. At least the demonstration worked. Okay. Oh my gosh, I was already worried that I have nothing to show you today. Yeah. Of course, you can. I've been thinking about some crazy things. I don't know. Maybe you're able by applying. I don't know, voltages in two different axes, X, Y axis. Maybe with a little joystick, you can actually make the cells go around. I don't know. Maybe some kind of a real life game type of thing where you can remote control the cells. Would be kind of fun to observe. Yeah, yeah. So, but basically. Wow, I have a lot of disinfection to do today. Clean everything with alcohol. Yeah, but yeah, it does seem to work a bit. No, not a bit, but it actually, yeah. The thing that I've learned here is that electrotaxis is not something only for Paramecium, but obviously also for other microorganisms, and evidently different microorganisms, yeah, move into different directions here. Yeah. What happens if you max out the voltage? You know what? Let's try that, because it doesn't really make a difference. If I end up killing the cells, who cares? So slowly start increasing. Let's slowly start increasing. Now they're moving all to the left, the tiny ones. So I've got maximum voltage is 30 volts. It's around. Yeah, they're still alive, still going now to the left. Let's see if I'm gonna reverse it again. Now they should be going to the right. Well, actually, now they're going to the right. They seem to be responding much faster at a higher voltage. Yeah. Okay. Yeah. See, now they're moving to the right. So yes, there is. Aha! Look at this. This one seems to be moving the other way. Okay. You know what I'm gonna do? This big one over here. Let's quickly reverse it. Let's see if it also responds to the electricity. The big one is now going the other direction. This big guy over here on the right side is now stuck somewhere. Keeps on moving. again, reverse. Let's see if it turns back. No, it turned back quicker than... Yep, no, you can't see it anymore. Okay, yeah. Wow, okay. Put salt into it, sodium chloride. Yes, because you can actually see that there is no current flowing. It's only voltage. Now they're all moving again. The tiny ones. Good. At least I'm happy that this demonstration also works without Paramecium, but also with other tiny microorganisms. So what I'm going to do now is... yeah, I'm going to do now the following. I will simply add a few, go through those questions. I've got a few questions here as well. I'm just gonna read them off. Yeah, here. There was this question I already answered. For feeding the Paramecium, I guess anything with starch would work. And it says, yeah, generally probably would work, but it's much more convenient to use the crushed starch grain, a wheat grain that I used. Yeah, PVA would also work. I talked about that. I was kind of worried that PVA, being water soluble, might dissolve, but then I was told that actually it's actually not soluble. Here the Paramecium going into the opposite direction. So actually, this seems to confirm that. Some cells move towards the positive side, some cells move towards the negative side. Yeah, do you know any substitution for Uparol that is compatible with alcohol? Honestly, I don't know it, unfortunately. Yeah, but I know that some companies, lab supply companies, do have certain mounting media that are much safer to use. Look, now here the whole swarm is coming. Look. All moving. Okay, I talked about the PVA glue. Do you know any substitution for safranine? Safranine is a dye. Generally, it is like this: the dyes react with different parts of the cell. Generally, it depends really what you want to stain. It's not like this that... sorry? Yeah, you can substitute any stain with any other one. But generally, if you need a general purpose stain, I recommend methylene blue. Or if you want to have something red, like safranine, you can try to use carmine red. Look, the Paramecium here generally moves, should be moving to the left. I'm bookbinding, and the teacher said that PVA is dry. You want to be able to, once PVA is dry, you won't be able to separate it anymore. Okay, so this seems to be confirmed that once PVA has dried, that it essentially, will be very stable. I want to get some new objectives for my microscope, but how do I know if they fit? Oh, that is a separate topic. Generally, it depends on the microscope that you have. If you have got a microscope with objectives with 160 written on it, then generally there's a large choice of objectives that you have. If you've got an infinity microscope, then you have to contact the manufacturer of the microscope, because different infinity microscopes have different standards. But generally, I recommend, if you're more interested in objectives, exchanging objectives, I have a second channel called Microbehunter Microscopy, where I actually talked about this. If you want all about microscope objectives and eyepieces and so on. It's not this channel, but the sister channel, so to say. Yeah. Is it normal for a current to be under 1 milliamp? I expect 8 volts to get much more current. So dirty water, and especially with such wide electrodes. Yeah, I actually also expected this. So the question really is why is there such a low current, right? And I did test this. If you actually touch the two electrodes together, then essentially, you have a current. Here it says a 0, which also surprises me a little bit, because but I might want to test it out. It could be maybe that the salt concentration is fairly low. Could you try introducing more microbes? Not now, because I've already done that, and it's difficult for me to reach it now. Okay. If you use a 4 times magnification, why use a cover glass? Well, generally because That's generally correct. At the low magnification, the thickness of the cover glass does not have a very strong effect. But generally, if you have a specimen that's not flat, and if you put a cover glass on top of it, then it will actually make the specimen flat, and it will also be easier to observe even with a under 4 times. And generally, you will not stay with a 4 times magnification, but you will actually go up as well. Yeah. So another paramecium going more towards the right side here. Yeah. I have dealt with indium tin oxide coated glass before. You might be able to etch the conductors into such glass coated with the conductive. There isn't, thank you. So a suggestion has been to etch the whole thing. I've just realized something. Maybe I could do the following. I have to ask a friend of mine if they have this possibility. It's possible to also, if you have a chamber, it's possible to vaporize metal and actually coat glass surfaces with metal. That would be actually quite nice. Then you would have then the electrode would be directly directly on the glass. You kind of basically just deposit a layer of metal directly on the glass surface. That would be actually, I think, the best solution, honestly. Yeah, I'll do a little bit more research here. So you know what? Let's reverse the whole thing again. I'm going to reverse the electrodes. Let's make them move again into the other direction just because it's so fun. So now they're not connected. Not connected. See, random movement. Random movement still. Look, did you see it's not connected? And then now I connect it. And off we go again. Now they're moving to the right. Yeah. So at least I'm a little bit relieved that the demonstration worked. But yeah. Single-celled organisms have no sense of pain because they have no nervous system and no sensors that transmit signals via nerves. That is correct. So how do they detect the increase in oxygen? Basically, what cells do what they have is a cell is not has a certain volume and a certain dimension. Obviously, see now the whole swarm is approaching again. And what you have is you have a concentration gradient of oxygen. Then one side of the cell will be exposed to a higher oxygen concentration than the other side because there is a gradient as oxygen diffuses. Yeah. Then there is a gradient, so to say. I'm just going to use this here as an example. Where's the arrow? Just want to show this to you over here. Where's the arrow? Here, let me see. Here we go. Yeah, you see this oxygen bubble here in the corner? This means that there's a high oxygen concentration here and a lower one over here. And this basically means that one side of the cell will receive or will sense a higher oxygen concentration than the other side. And this is how it actually, a lot of cells determine concentration gradients. Yeah, and how they're able to move towards certain, let's say, chemical markers, chemicals that they will detect, pheromones, as we would call them for higher organisms, right? But essentially, this is because chemical gradients, as they go from a higher to a lower concentration, basically also will make one side of the cell experience a higher concentration than the other side. And then there is basically a change then in the also I think of membrane potential that's and this also determines the movement of the cilia and so on. Yeah, so they are able to detect this without actually having nerve cells. Yeah, what could the evolutionary advantage be to be able to detect the electric field? How do they use this in nature? That's a good question. There is something, and every time when I do not know the answer. Because maybe there is no evolutionary advantage, because in nature maybe there is no electrical fields. It could simply be the following, that this is simply an effect how an electrical field can actually change certain membrane potentials around. And the membrane potential is again very important in controlling the movement of the cilia. So it could simply be that there is, even though there is no evolutionary advantage, because there is no evolutionary disadvantage, it simply is there, and it's simply an effect that we're able to observe. Yeah. There is also some bacteria are able to detect magnetic fields, so not electricity, but magnetic fields, magnetotactic bacteria, and it is assumed that those bacteria are able to this way sense up and down in a water. The reason is because the magnetic field lines on Earth, they will actually at the poles will be more vertical. So this was, I think, something that I also read somewhere, that the magnetic field lines of the Earth can actually be used for certain microorganisms to actually move into certain up and down in a column of water. Yeah? Because the magnetic field lines are not always parallel to the Earth's surface, but actually will also go into the Earth. Yeah, so this is something that I also once read. So there might be an evolutionary advantage for that. Yeah, but honestly, I do not know the evolutionary advantage, if there is one at all, concerning the ability of those microorganisms to follow electrical fields. Yeah. How do I get the chlorine out of the water so I can put the moss without killing the microbes? Quite easy. What you do is you let the chlorinated water stand open for a few days. The chlorine will evaporate on its own. Make sure that you have a water container, keep the lid away, and allow the chlorine to evaporate. Having said that, some microorganisms are very sensitive to chlorine. So you've got to be really sure. Yeah. Otherwise, just use drinking water. Carbonated water, you have to let it stand to remove the CO2, but you can also use a regular drinking water. Bottled water is sometimes also very good for growing microorganisms. Yeah? A physical model for galvanotaxis and a paramecium cell. Thank you very much. Okay. There is actually here somebody's done some research here concerning the galvanotaxis. Yeah. Off topic, where did I get that sample? This sample over here of Paramecium I made myself. Just the video that I published just recently in this channel actually shows you how I've done that. I simply took some pond water and I added a few wheat grains, and those wheat grains are then the food source for bacteria and also for other microorganisms. Yeah. And that's basically what I've been using now. Yeah. So, what time is it? Again, almost over. It's just so fun switching it back and forth. Let's go and make them go again into the other direction. Yeah. See, now some other cells are starting to move again. Quite interesting. Yeah. You could try magnets to see if magnets influence the organisms. Yes, I would have to get some very strong neodymium magnets. I know that some people are actually using magnets to collect magnetotactic bacteria. I was not successful in doing that yet. Yeah. But that would be indeed a possibility if you have a complex water sample. When I say complex, one with many different microorganisms, then it's possible maybe that some of them respond. Yeah. So because the biodiversity is quite high. Yeah, see those Paramecium over here, those few Paramecium generally, this one is going now more to the left, seem to be always going into the opposite direction of those smaller cells. Yeah. So, okay, at least we got that, got that to work. Yeah. Yeah. It's a little bit unfortunate that I'm not able to go all the way to the end, because I'm bumping into the Yep, here we go again. Yeah, the larger ones going to the left, the smaller ones to the right. Sometimes you're also able to see generally a movement of microorganisms also in an oxygen gradient. So that means that when you've got some specimens on a microscope slide, then sometimes you can also see that all of a sudden they start moving into one direction or accumulating in one part of the slide where there is the most oxygen. Yeah. Okay, but here there are 3 Paramecium now going to the left. Let's see very quickly if I can make them go the other way now. Yep, now they're going the other way. And let me reverse this again. See if I hope that we're able to catch them again. Here we are. Now they're going again to the left, 2 of them. Here more of them going to the left. Left. It's the smaller ones to the right. And let's give it a try again. Let's reverse. Reversed. Maybe I can make them go around. Yes, now they're going to the right again. Okay. Now they're going to the right, and now let's reverse it again. I think having some kind of a switch would be easier. They're going now to the left. Again, I hope. Here we go. So, wow, I have to tell you, I am now really, really relieved. They seem to move in a curved stream. Yes, this is the thing over here, right? You sometimes can indeed see that they also follow certain oxygen concentrations. Yeah, so sometimes you actually see them accumulate in a certain region, yeah, where there is most oxygen. Yeah. Wow. Okay. I'm kind of a little bit relieved that at least this demonstration seemed to work. For those of you who joined late, a short summary of what we've been doing. I've been the first part of this stream. I've been kind of not quite successfully experimenting around with some of those electrodes and some mishaps. But I think that's also maybe part of the whole thing, part of the fun. And then I now resorted to a commercial solution, which basically gives me a little more stability and control for the sake of simply demonstrating it. Those homemade do-it-yourself solutions definitely need to be optimized better, right? But I just wanted to simply give you also some kind of inspiration and trying out things yourself. Maybe you have better possibilities here. And if you take a couple of 9-volt batteries and connect them together in series, then you should also be able to get the sufficiently high voltage here. Yeah. So I think what I'm just going to do now is I think I'm going to call it quits for this week, for today. Start cleaning up the table here. In case you're wondering how my desk looks like. Where is the desk view? So that's basically my work environment here. Yeah, see, it's over here the water sample. Yeah, everything here, and there's also a whole range of things here over there on the side. Yeah, trash can for old slides. Yeah, and more water samples over there. Trash can for old pipette tips. Here, that's my monitor, and over there, of course, upside down now because I flipped the camera around, of course, the microscope, and over there is my stereo microscope. So I've also given you now a very short tour of my work environment here. I think I'm just going to call it a day for today. I'm kind of happy and relieved that the demo did work out after all. And yes, if you have any comments or questions or any suggestions maybe that you would like to share, the video of course is going to stay online, and there is a comments section also below where you can also share your own experiences. But for today, I'm going to leave it as it is. I wish you all the best. Happy microbe hunting as always, and see you around next time. Bye bye.